So this chapter is all about solutions, um, and why things will mix with other things, why things are soluble in other things, and the different things that affect solutions. You can't hear me? I think other kids can hear me. Sorry. Okay. Um, Maybe they accidentally pressed mute. Um, okay, so solutions. So let's flash back to what it means to be a solution. So remember, a solute, for example, is something that gets dissolved, and a solvent is the thing that does the dissolving. And if you take a solute and you mix it with a solvent, you have a solution. If the solution is called aqueous, it's because the water is, specifically the solvent is water. And now I know we talk about water being the solvent because it's obviously the most accessible and it's the one we have them, very readily and a lot of. But water is not the only solvent that exists on this earth. There's tons and tons and tons of other solvents people use in different other aspects. So just, I know we're used to using water. Just know that water is not like the only solvent. It's pretty universal, but it's not the only one that exists. And then something we haven't gotten much into has been this concept that you can have, right, we're used to like solids being a solute, but gases and liquids can also be solutes, and we'll talk about that more a little bit later. Okay, so again, what's going to cause a solute to dissolve in a solvent, or what's going to cause things to mix, is going to be based on their different types of intramolecular forces, which is what we focused on last chapter. And these are some of the types that we will kind of discuss. So the solution process involves two things, and I'm kind of jumping the gun a little bit, but I do want you to know that enthalpy, remember, is delta H, and you don't know this yet, but entropy is called delta S. And in nature, anything, not just chemistry, things tend to want to release energy, lower their energy as much as they can to achieve stability, meaning exothermic processes are favored. They tend to be the ones that happen spontaneously. Now, that does not mean endothermic processes don't happen on their own. They absolutely do, and there are plenty of endothermic processes in nature that happen on their own. However, exothermic is definitely favored and definitely happens more often. Exothermic processes where energy is being released and given off to achieve stability is definitely more common in nature than endothermic processes. Okay, so that's what this is saying. What this down here says is a little bit more into thermodynamics, which we haven't gotten into. We will next semester. And it's this idea of randomness, that things tend to become more disorganized or random, like on their own. And that's called entropy. So entropy tends to increase on its own. Meaning randomness tends to happen like without any help. So think about like if you go spray perfume in the corner of a room, it's not just going to sit in the corner of the room and like stay there all organized in the corner. Yeah, you sprayed it in the corner, but nature tends to want to, be randomized in order to achieve stability, so that perfume will eventually diffuse throughout the room on its own spontaneously and move around randomly. Another example I like to use for this is like cleaning your bedroom, right? Like, right, it seems to be that your room is like always a mess, right? Or at least my room is. Maybe your guys aren't, but right, your room's a mess. It's like you have no idea how it got that way. It just like randomly got disordered all on its own. And then in order for you to make it clean, you got to put in a lot of work, right? That doesn't just happen on its own. You have to like put in effort to get it to be clean, and then the next thing you know, it's like all messed up again. So it's not your fault. It's nature's fault for wanting to increase entropy. Don't tell your parents that though, because I don't want to get in trouble for you not cleaning your room because of nature. Okay. So Separation of mixtures. So we have these three different types that we're going to discuss. Filtration is one you're probably familiar with. Distillation and chromatography, probably not as much. So we'll do filtration first. So remember, filtration is to separate what kind of mixtures, and that's heterogeneous mixtures, meaning things that are not the same throughout. So you maybe have like a liquid portion as well as a solid portion. So if we were to like label this is a glass stirring rod. Okay. This is your solution. Well, I don't want to call it a solution. This is your mixture. Okay. This is a funnel. And then wrapped in the funnel, you have filter paper. So the filter paper catches the solid portion, while the liquid portion passes through, and that liquid portion has a name. It's called the filtrate. It's what you have after you filter. It's called filtrate. And so this is a way to separate a solid from a liquid, or a solid from some sort of solution. Okay? And this is filtration is used for heterogeneous mixtures, which is not what this chapter is about, right? This chapter is about homogeneous mixtures, also known as solutions. So solutions are homogeneous mixtures, meaning they're evenly mixed. So filtration, you cannot separate out homogeneous mixtures by filtration. It's not possible. So we have different techniques to kind of separate these solutions or these homogeneous mixtures. So a perfect example of a homogeneous mixture or a solution is salt water. You take salt, you dump it in water, it dissolves, and now you have salt water solution. Or sugar, you take sugar, you dissolve it in water, you stir it, and now you have a sugar water solution. So we are going to try to separate then these mixtures by changing or messing with applying methods that that can change our intermolecular forces. So things like boiling, for example, is what we're going to do. So we're going to look at distillation first. So distillation is a way to separate different substances. Now, you can have a solution that has maybe a solid dissolved in it, and you could separate it through distillation, or you could have two liquids. For example, like ethanol, which is drinking alcohol, and water. Ethanol boils at 78, water boils at 100. So if you can get the ethanol to escape sooner, which you would, you could collect the ethanol and then keep the water in the other container. So here's kind of the first example that they gave is salt water. So this is salt water, right? And then you heat it. Now this is stoppered, meaning nothing can escape. Okay. Now, if you wanted to collect the salt, you would just heat it, get rid of the water, and the salt would remain at the bottom, right? But what happens if you want to isolate the water? Well, this is called distillation, where you take and you heat up this solution, okay? And then you know that the water will eventually boil once you hit 100-ish, right? It'll start to boil, which means the water turns into the vapor phase. And then it starts to travel through this condenser. Now, this condenser has two parts of it. It has an inner part and an outer part. The inner part is where the water vapor travels through, and the outer part is being flushed with cold water. So you have hot water gas come in through here, and then it cools down in here because the cold water is being pumped through. And then so guess what happens when it cools down? It liquefies. It condenses back into a liquid. So you get it really hot, and then you cool it down, and you end up with pure water over here. And then at the bottom of the this flask, this boiling flask, would be salt. Okay. So the same thing is true if you were to do like ethanol and water. You would have a thermometer in here, okay, that would be measuring the temperature. And we know that ethanol boils at about 78 degrees Celsius. I think that's what it is. And so you have ethanol water mixture solution. And so you heat it until it starts boiling at 78. So then at 78, when you start seeing liquid drop down here, you know that's the ethanol because water doesn't start boiling till 100, right? So the ethanol will come out at 78, right? And then eventually it will stop once all the ethanol has been vaporized and then recondensed. You'll see this stop dripping. Meaning, once it stops dripping, as long as you haven't gotten to 100 yet, this should be pure ethanol and this should be pure water. Now, if you keep boiling to 100, eventually you'll boil off all the water too. But again, that's a way to separate a once homogeneous solution where filtration won't work. So pumping in the cold water does not mess with the measurements over here. So the temperature would change down here, right? This will be like cooler, I guess. I don't know how cool, but the water being pumped in, it's being pumped in just in this part right here, so it's not going to affect this flask. This is where you're caring about the temperature. Okay. So distillation is a process. And the other kind of process we're going to talk about for separating solutions is chromatography. So in chromatography, there's two parts. There's a stationary phase and a mobile phase. And we're going to talk about this more in a second. And there's a handful of types of chromatography, but we're going to talk about paper chromatography. Okay. So chromatography involves a moving phase and a stationary phase. So if we're doing paper chromatography, the paper is the solid or stationary phase, the piece of paper, okay? And then the liquid that you dip the paper in is called the mobile phase. It moves. Why? Well, because we know liquids tend to climb up paper by capillary action. So they call it the mobile phase because the liquid will move up the paper. Now, how this works is usually the paper has one type of polarity and then the solvent has a different type. So like maybe the paper is polar and the solvent is nonpolar, or the solvent is polar and the paper is nonpolar. How far the mixture you're focusing on travels up the paper depends on how attracted it is to either the paper or the solvent, which will tell us basically its polarity or its affinity to that substance, as long as you know whether the paper is polar or nonpolar and whether the solvent is polar or nonpolar. So again, they use this using paper chromatography. And the example we're going to use, well, the example I'm going to show you in a second, our solvent is going to be water. So that means our solvent is our polar species, our mobile polar species, which means the paper is going to be non-polar. Okay. And again, you take your mixture, and this is often used for like dyes and things like that are homogeneous mixtures. You take your mixture and you draw a line on your chromatography paper and you put that dye or that mixture, or I'm going to show you an example of a marker, a washable marker right here, okay? And then you let it run, meaning you dip the paper in the solvent, in this case it's water, right? And the dye breaks apart into its different components. So a separate component, and then a separate component, okay? So the dye breaks apart into its components at different locations based on its affinity to either the solvent or the paper. So what you can do, and we don't do this anymore, but I do want to talk about it, is you can measure the RF value, the retention factor, based on how far each one travels. That's what the top number is, how far it traveled, and you measure it like with a ruler, like millimeters, divided by how far you let this run before you stopped. Where your solvent front ends is where you stop the experiment, because you don't want it to run off the paper. And that's called your RF value. So based on this chromatogram, that's what this is called, whoever the solvent is, this guy had the most attracted forces to the solvent. Why? It traveled the furthest with the solvent, so it must have been more heavily attracted to the solvent. Whereas this guy, R, was more attracted to the paper than it was the solvent. It didn't travel very far. It would rather stick to the paper. So it had a higher affinity to the paper as opposed to the solvent. And that's how you read these. So if you look at these bunch of different real life developed chromatograms, you have different markers. I think this was black, washable marker. This was brown. this was red, this was green. This is not blue. I don't know which one this is, but it's not blue. And then this one was like orange, right? And what they did, so they drew the line and they just put dots of the ink, which is a solution. It's a homogeneous mixture of ink, right? You go and you color on your paper with a Crayola marker, a Crayola black marker, right? It looks homogeneous, right? This is a homogeneous mixture. However, it's actually made up of a bunch of different dyes. And so they use this chromatogram to separate it out. So look at the different dyes that make up your black marker. And look at the different dyes that make up your brown marker. And look at the different dyes that make up your green marker, and so forth. Okay, so chromatography separated out a solution that we couldn't separate out, obviously, by filtration, because it was all one component. Evenly mixed one component. So look at this one. So you have like the red, and then you have the orange, and then you have the yellow, and then you have this like almost like a lavender color, and then purple. So all of these dots right here have specific retention factors. It is very apparent that this dot was the most attracted to the mobile phase, where the red dot was most attracted to the stationary phase, or the paper. This is a real-life chromatogram of the chlorophyll, right? So chlorophyll is a pigment that we know that's in plants, okay? And it's a pigment composed of multiple different parts. Now, it all looks the same, right, if you look at it without separating it, but it does have, the pigment does have different parts. It is composed of different things. It is a mixture, okay, of different colors that give it the overall green color. So yeah, the attraction to if the paper, well, the paper isn't always nonpolar, as you're going to see in this one. The paper can be polar, but the reason anything would be attracted to something that's nonpolar is because it itself is nonpolar or has dispersion forces. Yes. So look at this FRQ, which was not that long ago on the AP. This was on the AP test. They give you chromatography chambers. The reason they do that is sometimes you don't want to breathe in the chemicals or the solvents. Also, some of the solvents can readily vaporize, so you want to kind of try to keep that from happening. So like this box right here is the chamber. Sorry. And then this right here is like your paper that they're dipping it in, your chromatography paper. Right? And then the solvent, whoever it is you put the tip of the paper in the solvent, okay? So they had 3 dyes, A, B, C, all right? They ran the experiment and got results, and then they did the experiment again with one of the dyes, but they didn't tell you who it was. It's either A or B or C, okay? And they run it again, and obviously they're gonna try to ask you to figure out who the unknown is. They tell you, though, that the solvent this time is nonpolar, which means it's obviously not water, right? Because water is polar, so it's some solvent that's nonpolar. A pretty common solvent that they used to use a lot in organic chemistry is carbon tetrachloride. It's a nonpolar solvent. Anyways, the solvent is nonpolar, so the mobile phase is nonpolar, which obviously then tells you that the stationary phase, aka the paper, is it has to be polar, right? So it says which dye, A, B, or C, is the least polar. So the most nonpolar is the least polar, right? So the most nonpolar is going to be or be attracted to the thing that is nonpolar, while the solvent is the thing that is nonpolar. So in this instance, the answer should be C. C traveled the farthest, meaning it was the most attracted to the solvent because it traveled the farthest with the solvent, and the solvent was nonpolar. So it's obviously the least polar because it was the most attracted to the thing that was nonpolar. Questions on 4A? Okay, what about 4B? It says which dye is present in the unknown sample? So here's the unknown. Now you need to be careful, right? Because where they started and where they ended are different in the chromatograms. So this is where that RF value would come into play. So let's just look at the unknown. I'm going to make up numbers in millimeters. To me, if I'm being honest, this right here, how far my dye traveled relative to how long this ran, is like, looks like perfectly half to me, right? Like 0.5. So maybe it traveled 2 millimeters and the whole distance was 4 millimeters to give me 0.5, right? It traveled half the distance, okay? So basically you want to pick the dye that did the same thing over here, that traveled about half the distance relative to the overall solvent front. So who did that? Which dye? A. Look, this looks like it went up half of the chromatogram compared to the whole chromatogram, right? So maybe it went up 3 millimeters out of a total of 6 millimeters. And I did more because, right, this obviously is longer than this one. But again, still the RF value is about half, which, so the unknown is very likely dye A. Okay? All right, so the two words that we're going to talk about, and they are used interchangeably sometimes, but they really don't mean the same thing. The word miscible is two substances that have the ability to mix together, but they don't change their identity, meaning they retain their same chemical and physical properties. So like if you take water and you say it's miscible in methanol, it means they'll mix. So like you can't tell them apart necessarily. However, the water's still the water and the methanol's still the methanol. Okay, they don't change who they are and they don't physically change. The difference between this and solubility or being soluble is if something is soluble, it actually generally changes physically the substance. So for example, if you take salt and you dump it in water, the salt doesn't stay solid NaCl. The salt changes formation into sodium ion and chloride ion. So yes, it's still salt, but it's salt in a different phase. Now they're aqueous ions instead of solid. So that's what it means to be soluble as opposed to just miscible. So we're going to go over the different types of solutions and then the factors that affect them, and that will be it for today. So liquid solutions, as I was just saying, methanol and water. Why are they able to do this? Well, because water's polar and can hydrogen bond, and methanol is polar and can hydrogen bond. So they're similar in terms of forces, so they can be soluble and miscible in one another because like dissolves like. Polar with polar, nonpolar with nonpolar. Now, I don't need you to know this number, 6-carbon chain. Eventually, your organic alcohols become too big to be able to mix with water. So yes, while all alcohols have this OH group, which means this H is capable of hydrogen bonding, the longer this carbon tail gets, the more nonpolar it becomes. And after 6 carbons, and again, you don't need to know that it's 6, but it is, after 6 carbons, basically the nonpolar region becomes so strong that it overpowers the ability of the alcohol to even be able to mix with water. There's just too much non-polarity in the, I usually call this like a tail, this hydrocarbon tail. To allow it to continue to mix even though it does have an OH group. Okay, so this is a liquid solution. So which of the following liquids experiences dipole-dipole intermolecular forces? I'm going to cheat real quick and tell you these guys' shapes. So it says, which of the following liquids experiences dipole-dipole intermolecular forces? AKA is polar. Things that are polar can have potentially dipole, or can have dipole-dipole interactions or intermolecular forces. Oxygen is nonpolar. It's out. Carbon tetrachloride, tetrahedral, nonpolar. It's out. Sulfur trioxide is trigonal-planar. Trigonal-planars are nonpolar. It's out. Now, CF2Cl2 is tetrahedral, but it isn't bonded to all of the same atoms. There is an uneven pulling of the electrons between carbon and fluorine bonds and carbon and chlorine bonds. So because this, even though this is tetrahedral, this is polar. There's uneven overall uneven sharing of electrons. There's a net dipole moment. So D would have dipole-dipole forces, meaning CF2Cl2 interacting with CF2Cl2 other molecules or any other molecules that are also polar. Okay. All right, this one says, which of the following molecules is the least soluble in water? Well, that's going to be D, because its hydrocarbon tail is the longest. So even though these are all alcohols and they can all hydrogen bond, this one isn't going to do it as good as the other ones because this is a super nonpolar part of the molecule. Okay. One of the most common types of solutions you guys are familiar with are solid liquid solutions, where you take a solid and it dissolves in a solvent. If that solvent is water, they're called aqueous solutions. And certain salts will have the ability to do this because of ion dipole interactions that allow polar solvents to break apart ionic compounds. That's how you make a solid liquid solution. Sugar in water, for example, though, sugar does dissolve in water and makes a sugar water solution. But that happens because sugar is polar and is capable of hydrogen bonding. Sucrose, it's not ionic. Table sugar is C12H22O11. It's an organic molecule, but again, it's capable of hydrogen bonding, so it readily dissolves in water for that reason, not for ion dipole reason. Sugar does not break apart into ions when it dissolves in water. Okay. And then again, remember in ionic ion dipole interactions and ions dissolving in water that the waters will orient themselves where the partially negative oxygen region is attracted to the positive cation of the salt. Okay, and it will kind of orient itself around it that way. And then the positively charged hydrogens will be attracted to the negatively charged anion of the salt. Okay, and it'll kind of orient itself that way, and these form what are known as hydration shells. But again, this is ion dipole interaction is what allows salts to be soluble in polar solvents. A gas liquid solution. So any of your carbonated beverages, it's a solution of liquid with gas. Most of them, for sure, a main component is water. If you have soda, there's also sugars and stuff in it, but again, it's water, and the word carbonated comes from the fact that carbon dioxide has been dissolved in the water. Okay, so that's a gas liquid solution. Oxygen in water. So this is like if you think about different bodies of water, lakes, rivers, streams, oceans. We know that there is oxygen in the water, and we know that because all animals need oxygen to live. And so we know that the oxygen, which is technically non-polar, is dissolved in the water, which is polar. And you go, well, how does that happen? If oxygen is non-polar and water is polar, how can you get oxygen to dissolve? Well, the really polar water induces the dipole on the non-polar oxygen, which is what causes the bodies of water to have oxygen dissolved in them. All right, now this is kind of probably a newer one, but I need you to kind of steer away from the idea that solution means something has to be a liquid or have a liquid or be wet. I know that when you think solution, like you instantly think like liquids, wet, right? But a solution is just an even mixture. So like the air you breathe is a solution of gases, okay? It's an even mixture of gases. So gas can be a solution. So let's talk about our air and the 3 most abundant gases. Who is number 1? Who is the most abundant gas in our atmosphere? No one knows?
Is it carbon?
No. Keep going. It is nitrogen. 78% mostly nitrogen. Okay, so it's our solvent. It's the thing we have the most of. Oxygen is next, coming in at like 21%. And then argon is the 3rd most abundant. Okay, argon is the 3rd most abundant. And again, it's a mixture. The air you're breathing is a solution, an even mixture of those gases. And yes, there's other gases mixed in there. There's, carbon dioxide and things like that. But nitrogen, oxygen, and argon are our 3 most abundant gases, and they are a solution of gases that we breathe. Okay, you can also have a gas solid mixture. We're not going to talk much about this, but where you have, like, for example, this is hydrogen gas embedded in between some different metal atoms, evenly mixed. Okay, and that creates a solution. You can have solid mixtures. So we know, like, for example, some alloys are solid solutions or mixtures. For example, steel has all these iron atoms, and then embedded in the spaces between them are carbon atoms. So iron is a solid, carbon is a solid, and again, evenly mixed, right? And this is what gives steel its super strong properties. It's more rigid because the carbon fills in these gaps, and the iron atoms then can't like slide past each other as easily, and that's what makes steel so strong. But again, this is a solution of solids. And then another example, so again, this was an interstitial alloy, whereas this one is a substitutional alloy. So like brass, a solution of brass, it's solid, happens when you have a bunch of copper atoms, and then some of the copper ones are replaced with zinc, and you make what's known as brass. Now, this doesn't just happen, right? This involves like melting, heating, mixing, and then resolidifying. But again, it's still a solution of solids, okay? And our solutions are discussed by having concentration, and there's tons of different ways to discuss concentration. Mass percent is something we've already discussed. Mass percent is the mass that, I guess we'll go into it. We'll go into all of these different types. Molality is no longer included on the AP test, but I am going to just briefly mention it. But remember, solutions will have concentration because they're mixtures. Pure substances do not have concentration. Like, you have to be a mixture to have concentration, okay? So let's have a flashback to what mass percent is. Mass percent is the mass of the thing that we're looking at, whoever it is, divided by total mass. And then to turn it into percent, we multiply by 100. Okay? We don't do much with this, but I did want to mention it to you. Parts per million or parts per billion is really mass percent, but instead of turning it into a percent, you multiply by 10 to the 6 for million, or 10 to the 9th per billion. And that gives you your parts, that number you'll just write as ppm. And again, you see this a lot in environmental science. They talk about like thermal and environmental pollution with like ppm and ppb, like carbon emissions and things like that. So you'll see like what's the concentration of carbon emissions in our atmosphere today. They may write those numbers in ppm or ppb. Again, we're not going to do much with this. I just wanted you to recognize it as an alternate way to discuss concentration mathematically. Okay. Mole fraction we've also done. Remember, it's the symbol X, and mole fraction is just the moles of whoever you're looking at over total moles. Moles over total moles. We've done this before. We did this in the gases chapter. Molarity was in chapter 4. Molarity is moles per liter. Moles of solute per liter of solution. Remember, solution is the solute and the solvent combined. Okay. Molality is moles per kilogram. Now, they don't have molality anymore on the AP test. And you should notice that the numerator is the same as molarity, it's moles of solute, but the denominator is kilograms, so it's mass, of just the solvent, so not of the entire solution. So molarity and molality are slightly different in terms of what they describe, but they both are units for concentration. Again, we're not going to do molality. I just wanted to show it to you so that you at least saw it and know that it is another type of unit for representing concentration of a solution. Okay, so I'm going to give you about 3-ish minutes to please do this problem. This is review. This is not new stuff. So I ask you to calculate the molarity, mass percent, mole fraction, and then you don't have to do molality. So please take a moment to work on these 3, and I'll get back to you when you're finished. All right, so I have kind of the worked out answers for you guys. I mean, the answers are on the slide, but here's how to do it. Do I have any questions on how to do molarity, mass percent, or mole fraction? Again, this is review. Any questions? Mass percent could be done with moles, but it's generally by mass. That's why they call it mass percent. Mass means grams, really, or kilograms. All right, so let's do one more practice. And I'm going to do mole fraction because that tends to be a commonly misunderstood one. So it says an aqueous solution of HCl is 36% HCl by mass, and I want to know the mole fraction. Again, we're not going to do molality, okay? 36% by mass, so what we do, percent means per 100, right? Sorry. It means per 100. So if this is 36% HCl by mass, we're going to call this 36 grams, right? So 36 grams of HCl. Well, if it's an aqueous solution, the other component is water. So how much of it's water? Well, the rest is water, right? So that would be what? 64 grams of water, because 64 plus 36 is 100. And then mole fraction is moles of the thing you want, which is HCl, over total moles. Mole fraction. So we need to turn grams of HCl into moles of HCl and grams of water into moles of water. So 36 grams of HCl. HCl is 36.06 grams per mole. Oh, I'm sorry, 36.46. Ooh, I wrote that wrong last class. Wow, nobody stopped me. So the moles of HCl is 0.987 divided by 0.987, because, right, it's part of the total mole, and then we need to know water's moles. 3.55. So HCl on the top, and then HCl and water mole on the bottom, and that's how you get 0.22. Do you have any questions? Okay, so hopefully that was a little flashback to concentration calculations, okay? And again, so this whole concept of this like dissolves like is that solutions that are similar in attractive forces or intermolecular forces will be either more soluble or miscible than things that don't like each other. So that it's this concept of like dissolves like. So nonpolar solute will be soluble in a nonpolar solvent. Nonpolars with nonpolars. And then ionics and polar solutes will be soluble in polar solvents. So polars with polars, nonpolars with nonpolars, but then polar and nonpolar would not dissolve, be soluble, or miscible. Okay? It's this concept of like dissolves like. So look at this container. We've got carbon tetrachloride, nonpolar. You've got hexane, nonpolar. Now there's a barrier, okay? But if you remove the barrier, this is showing, like this was blue, this is red, now it's purple, purple kind of indicating that they've obviously mixed, right? So this is kind of that concept of entropy as well, right? If you remove the barrier on their own, these molecules are going to be more disordered. They're going to move around randomly. They're not going to just stay on their side. They're not going to stay ordered in their own little corner because things tend to want to move around randomly on their own. Now, why are these allowed to do that? Why will they mix? Well, they will mix because they're both nonpolar, and nonpolar things are miscible and or soluble in nonpolar things. Now, when you take a solid and you put it in a solvent, okay, just because the solid disappears does not mean it dissolved or that you made a solution. Sometimes the solid disappears because you've had a chemical reaction. So just be careful. Just because a solid goes away when you put it in a solvent doesn't mean you've formed a solution. You could have caused a chemical reaction, okay? So what affects solubility? So it's, again, this is what we've been talking about. This concept of like dissolves like, polar with polar, nonpolar with nonpolar. We're also going to talk about pressure, which is going to be how more gases can be soluble, or how gases can be more soluble, and then temperature. Okay, so again, the nature or how structures are organized, meaning like, can they hydrogen bond? Are they polar? Are they nonpolar? Those interaction determines how soluble something is. The more polar something is, the more soluble it is in something that's polar, okay? Now, gases in a solvent, the solubility of gases in a solvent increases for two reasons. One of the reasons is the heavier the gas is, the more dispersion forces it has. And the more dispersion forces it has, the better of a chance it has of interacting with something that's potentially, if it's polar, okay? Or the more polar the solute, the gas is, the more polar the gas is, the better of a chance it will be dissolved or interact with a polar solvent like water. Okay. So again, miscible are liquids that mix. Immiscible means liquids do not mix. Okay. Hydrocarbons are heavily nonpolar, meaning just carbons and hydrogens. They're super big long chains of just a bunch of nonpolar, dispersion forces only. However, some organic molecules are polar, like our alcohols, right? But the longer that carbon chain gets, the less soluble the alcohol becomes. So I have a test tube where I have 3 different chemicals, okay? It's obvious that the carbon tetrachloride is more dense than water, whereas hexane is less dense than water. But water is forming this like barrier in between the layers. There's 3 layers. Anybody want to care to explain why there's 3 layers? It is because water is polar, yes. And what are hexane and carbon tetrachloride? This is a hydrocarbon, so it's nonpolar. This is tetrahedral, so it's nonpolar. So look at the water separates the 2 nonpolar substances from each other. Carbon tetrachloride and water aren't going to want to mix. One's polar, one's nonpolar. Water and hexane aren't going to want to mix because water's polar and hexane is nonpolar. So what would happen if you shook the test tube? How many layers would there be if you shook the test tube? 2. Good job. Why? Because if you shook the test tube and this guy had a chance to interact with this guy, well, nonpolar and nonpolar things will be soluble in one another, right? So like this layer and this layer would combine, and then the water would separate out. Okay? Again, it's this concept of like dissolves like. So this question says, will hexane, which is nonpolar, or methanol, which is polar, be the appropriate solvent for grease? So grease is obviously nonpolar. It's a hydrocarbon. So hexane would be the better solvent. What about potassium iodide? Well, potassium iodide is ionic, and we know ionic salts are soluble in polar solvents because of ion-dipole interactions, right? So methanol would be the better solvent for potassium iodide. This question says to predict whether each of the following substances will dissolve in carbon tetrachloride. So carbon tetrachloride is nonpolar. Okay, so if it's going to dissolve in it, you're going to want to have something that is also nonpolar. So this is heptane. Hydrocarbons are nonpolar, so yep, this would be a good one. Okay, sodium sulfate. This is ionic. Ionic compounds are not very soluble in nonpolar solvents, so nope. HCl is polar. Carbon tetrachloride is nonpolar. Nope. Iodine, nonpolar. Whoop. It would have a good chance of dissolving in something that's nonpolar. Okay. So I do say name them, but I don't actually need you to name them. I am going to name them for you. If you haven't noticed, organic molecules, carbon hydrogen chains, can get ridiculously long. So what organic chemists do is this line notation or line method, where instead of drawing all the carbons and the hydrogens out, you just see all these lines and assume that they're there. And at every point, it's assumed to have a carbon. So anytime you see a point, there's a carbon attached to it. And then it's also assumed that the appropriate number of hydrogens are attached based on the notation. So it's just assumed. All the carbons and hydrogens are just assumed to be there. Okay, so carbon, 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 carbon. All right. Now, let's name it then. 1, 2, 3, 4, 5 carbons and an OH group. So this is pentanol. This is 1, 2, 3, 4, 5 carbons, nothing else. So this is just pentane. 1, 2, 3, 4, 5 carbons, and this is a halide, halogen. They'll call this chloro because it's chlorine, right? Pentane, because there's still 5 carbons. And this one is called 1, 2, 3, 4, 5. 1, 5, pentadiol. 5 carbons, 2 alcohols on carbon 1 and carbon 5. Crazy, huh? Anyways, that's not the point of the slide. The point of the slide is to arrange these in order of increasing solubility. So the least soluble to the most soluble. Well, pentane is the least soluble. It's a hydrocarbon. It's nonpolar. So this guy's out. This guy should be fairly soluble because it's going to be polar because of this chlorine on the end. So it has dipole-dipole forces. But it's not as soluble as this guy because this guy can hydrogen bond. And hydrogen bond generically is stronger than just a plain dipole-dipole force. So why is this guy the strongest, though, or the most soluble? Well, this guy has the opportunity to hydrogen bond more than once, therefore increasing its solubility. Okay. All right, let's talk about vitamins. There's tons of different types of vitamins, but let's talk about just 2 different ones. So vitamin K is a fat-soluble vitamin, and if you did not already know this, fat is nonpolar, okay? So you don't need a lot of vitamin K daily in your diet. Why? Because it gets, dissolved and stored in your fat, and so it's just kind of there where you're not like getting rid of it. So if you look at vitamin K, it's a huge long chain, hydrocarbon chain, which is super nonpolar. And then you go, ooh, this kind of like gives us a chance, right? To think, ooh, there's a pull here, right? A polarity, but then there's an opposite polarity down here. So this is super nonpolar, all right? Which is why it's so soluble in fat, which is also nonpolar. And because it gets stored in your fat and not like excreted, you don't need a lot of it daily. Now, the opposite is true for vitamin C. So check out vitamin C. Vitamin C is water soluble. Well, we know water is polar, so obviously vitamin C is polar. Look at this guy. Look at all of these abilities of vitamin C to hydrogen bond to water. So vitamin C readily dissolves in water, which is why you have to have a lot of vitamin C and you have to replenish it every single day, because your body's constantly absorbing it, dissolving it, and then getting rid of it. How do you get rid of water soluble things? How are we getting rid of the water soluble substances? You don't need to be afraid to share. How do you get rid of this? What do all water soluble substances, how do we get rid of them? How do our bodies get rid of them? Pee it out, right? You pee it out, right? So because it's constantly doing that, you constantly need to be replenishing it, as opposed to the vitamin K. So, right, this is chemistry in everyday life of why you need more of certain vitamins than you don't of others. Okay, the solubility. Okay, so let's talk about solubility and pressure. Now, pressure is only going to affect the solubility of gases. Okay, so remember that if we change the volume of a container, we can change the pressure. And there, you used to have to do calculations with Henry's law, which is down here. But all Henry's law is saying is that pressure of a gas and the concentration of the gas dissolved in the liquid, usually, or I guess all the time, depends on, or are directly proportional. Meaning more pressure, you can get more concentration of gas dissolved. Less pressure, less concentration of gas dissolved. So let's look at what that looks like. You have this container right here. There's a liquid right here, and we do see some gas molecules dissolved, right? So this has a concentration of gas in the container. Well, if we move this piston down, so we decrease the volume, therefore increasing the pressure forces more of the gas molecules to dissolve in the liquid. So you have a higher concentration of gas dissolved in the liquid when you have higher pressure. It's a direct relationship. Lower pressure, less gas concentration dissolved. Okay? So now check out this guy. This is just showing 3 different gases being dissolved specifically in water, okay? And of these 3 gases, hydrogen is the least soluble, okay? And why is that? Well, it's nonpolar and water's polar, so it really doesn't want to be soluble in water for that reason. And you can increase the pressure to help it be a little bit more soluble, but it like only does so much. And then oxygen is next. It's also nonpolar, but it's more polarizable, right? It's heavier. So it has a better chance of being soluble in water. And then the more you increase the pressure on it, the more of the oxygen that can dissolve in water, higher concentration. NO of these 3 has the most solubility in water because NO is polar. And water is polar. So this like dissolves like, right? And then again, as you increase the pressure, you can get even more of it to dissolve. Okay, temperature is probably something that you're fairly familiar with in terms of solubility for solids. So if you take a solid and you dump it in water and then you heat it up, when you heat it up, those particles move faster. When they move faster, they can break apart easier, right? So generally, as you increase the temperature of a solid in a solution, you can increase the solubility of that solid. So if you heat it up, you're able to dissolve more solid and more solid and more solid. The more you heat it up, you can get more solid to dissolve because now the particles have more kinetic energy. The problem with that with gases is it's the opposite concept. If you heat up gases that are dissolved in a solution, the gas molecules now have more energy, just like they do in the solid phase. But when they have more energy, they're going to escape out of the solution into the gas phase. So you actually decrease solubility by increasing the temperature of solutions that have gases dissolved in them. So we're going to talk about this in relationship to thermal pollution and soda. So first we're going to talk about thermal pollution. Thermal pollution is pollution caused by heat. Okay, that's what the thermal part means. So let's talk about nuclear reactors. What do you know about where nuclear reactors are always located next to? Anybody? Nuclear reactors are always located next to a body of water, okay? Or almost always, I guess I should say. And why do they do that? Well, that's because like the nuclear reactions, right, that we use to get energy are highly exothermic. They get super duper 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 hot, okay? And they get so hot that if we didn't cool them down, right, the nuclear reactor would melt down and that would be problematic. Yes, right? So what do? Well, they pump in the cool water from the body of water and to kind of cool the nuclear reactor, and they pump that water back out. So the cold water comes in, but then they spit warm water back out. So why is that bad? Why is this considered thermal pollution of the water? Why is that bad? Why can't you just be like, oh, I don't care, it's warm water, who cares? It totally does change the environment for any other plant or animal life living in there. But why? If you had cold water and then you're dumping warm water back in, what do you know about warm water? Warm water has less oxygen dissolved in it. What do the plants and sea life need? Oxygen, right? By heating up the water, you end up with less oxygen dissolved in the water. Less oxygen is not good for any sort of plant or animal life that needs it to survive. So that causes thermal pollution. Okay. So we're going to talk also about Henry's law. This is pressure and concentration, right? That's his law for gases, and we're going to think about soda. Now, this whole slide is going to say everything I'm going to say, so I'd rather just like look at the picture in a second. What happens when so Coke is my favorite soda. I don't drink a lot of it anymore. Also, McDonald's Coke, oh, so good, okay? But what happens when you open, so soda is carbonated, right? So how do they get soda to be carbonated? Well, they take and they pump carbon dioxide in there, and they increase pressure to get more carbon dioxide in there, and then they seal it, right? So what are you hearing the second you hear the noise when you open it? What on the molecular level is happening when you hear the from opening a can of soda or a can of any carbonated beverage? It doesn't have to be soda. On the molecular level, when you hear that CO2 is escaping, absolutely. The carbon dioxide is jumping out. Why? Because you lowered the pressure, right? By opening it, you lowered the pressure, so some of it's like, see you later, I'm out of here, right? Now, luckily, it doesn't all just escape at once, or that would really suck unless you like flat soda. It doesn't just all escape at once, and usually most people are pulling it out of a refrigerator, so it's also cold. But then after you leave it out for a while, two things happen. One, if the container remains open, which it does, the can stay open, right? Gas molecules will escape out. What is the driving force of that over time? Why does soda go flat over time? Again, this is chemistry based. Why does soda go flat over time? So, right, there's not as much pressure keeping the molecules in, correct? and also it gets warmer, right? So the carbon dioxides over time get warmer. And if they get warmer, they're going to escape, which is why your soda eventually goes flat. That and it's open, the container's open. So if you open it and you take a sip or whatever, what would be a way you could keep it from going flat, like in a let it stay carbonated longer? How could you prevent it from going flat as fast? Like what could you do to kind of keep it carbonated longer instead? Now, this isn't obviously eventually it will go flat as long as the opening is the container's open, because all of the molecules will eventually escape out of the opening. But how could you slow that process down of it going flat? Okay, sealing it would be one way, but the easier way would be like throwing it back in the fridge if you aren't finished with it. It will take longer to go flat if you keep it cool, because the gas molecules won't escape as quickly. So if you keep it in the fridge longer, even though it's open, it won't go flat as fast. Okay? And again, this is all this Henry's law idea of concentration of the gas, and it depends on the pressure and then the temperature. If you were to put an open can soda in a sealed bag, it should slowly inflate, assuming you aren't losing anything to the surroundings. Yes, because the carbon dioxide gas should be able to be collected. Okay, so again, this is kind of this visual. And this is what I'm ending with. So here we are. We are fishing chemists, okay? So my question to you is what is the point of this diagram? So it says this person studies chemistry, and it's apparent that they have a longer fishing rod than the person that does not study chemistry, meaning the longer fishing rod, it looks like this guy's going to catch more fish, which I guess is ideal, right, if you're trying to fish. So what do you think this is trying to say? It looks like the fish are down here, right? And there aren't fish up here. Why do you suppose that is? So great, right? The fish, the surface of the water is going to be the warmest, right? Because that's where the sun is hitting it. So this is like the warmest. What do you know about the warmer water? Well, there's less oxygen dissolved in warmer water. Whereas down lower is cooler water, right? Which is more oxygen dissolved. So the fish would be happier down here where there's more oxygen dissolved. Does that make sense? All right, so this is where we're ending today.